رساله الدكتوراه بتاعتي اللي تمت مناقشتها من حوالي اسبوعين. عنوان الرساله Effect of Adenosine Receptor Antagonist Niacin and Curcumin as Neuroprotective Agents Against Parkinson's Disease Induced by Routine Illness. This is submitted for the Doctor of Philosophy, degree in Pharmaceutical Science Biochemistry, presented by Rashad Ahmed, Assistant Researcher of Biochemistry, Therapeutic Chemistry Department, National Research Center, supervised by Dr. Tariq Kamel Mutawa, Professor of Biochemistry, Faculty of Pharmacy, Cairo University, Dr. Nermeen Abdelhamid Sadiq, Professor of Biochemistry, Faculty of Pharmacy, Cairo University, Dr. Manan Abdelaziz Khalid, Professor of Biochemistry, Head of Pharmaceutical and Drug Industries Research Division, National Research Center, Dr. Sanay Ahmed Ali, Professor of Biochemistry, Therapeutic Chemistry Department, National Research Center. This study was published in Molecular and Cellular Biochemistry Journal, Impact Factor 2.3. Introduction. Parkinson's disease is a chronic neurodegenerative disease associated with substantial morbidity, increasing mortality, and high economic burden. Incidence of Parkinson increases with age, but an estimated 4% of people with Parkinson's disease are diagnosed before the age of 50 years. Men are 1.5 times more likely to have Parkinson's than women. The disease is described as a shaking palsy by James Parkinson in 1817. Jim Martin Chaput reports its current name honoring James Parkinson. Parkinson also called idiopathic or primary Parkinsonism, hypokinetic rigid syndrome, and paralysis idiopathy. The recognizable symptoms of Parkinson's disease are just the tip of iceberg, such as tremor, rigidity, and bradykinesia. Other symptoms include constipation, loss of smell, hypotension, sweating, hallucination, anxiety, and depression. This figure shows dopamine in healthy and Parkinson's patient. In normal neurons, there is normal level of dopamine. When 80% of dopamine produced in cell are damaged, symptoms of Parkinson's disease appear. Motor symptoms of Parkinson: forward tilt of trunk, reduced arm swinging, shuffling gait with short steps, rigidity, and trembling of it. Parkinson's disease is characterized by the loss of dopaminergic neurons in the substantia nigra. The substantia nigra is a basic ganglion structure located in the midbrain. This plays an important role in reward and movement. Substantia nigra is Latin for black substance, reflecting the fact that parts of the substantia nigra appear darker than neighboring area due to its high level of neuromelanine in the dopaminergic neurons. This located of substantia nigra in the brain. This figure shows healthy dopamine production in substantia nigra and cell degeneration and decreased dopamine production in Parkinson's disease. Hippocampus is the earliest and the most severely affected structure in several neuropsychiatric disorders such as Parkinson's disease, Alzheimer's disease, epilepsy, schizophrenia, and depression. It is located hippocampus in the pathogenesis of Parkinson's disease: free radical and deficit in energy metabolism, programmed cell death, genetic factors, environmental factors, protein aggregation, aging, and drug-induced Parkinson's. The ubiquitin pathway and the mitochondrial pathway are the two main pathways involved in Parkinsonism. The ubiquitin pathway toxicity arises from alpha-synuclein misfolding protein and aggregates. These proteins are degraded by ubiquitin proteasome system when accumulation or failure of clearance by ubiquitin proteasome system, leading to formation of Lewy body. The mitochondrial pathway, there is loss of mitochondrial potential membrane, leading to opening of mitochondrial permeability transition loop and release of cytochrome C from inner membrane of mitochondria to cytosol, which cause activation of caspase and cause cell death. Dysfunction of both pathway leading to oxidative stress and cell death. Most of the knowledge of therapeutic progress in terms of Parkinson have mainly come from different animal models that have been developed in order to reproduce human Parkinson. Models used are pharmacological induced model, toxin induced model, genetic models, protein induced Parkinson rat model is used in the present study. Protein is an odorless, colorless crystalline isoflavone used as a broad-spectrum insecticide and pesticide. It occurs naturally in the seeds and stems of several plants, such as the jicama pine plant and the roots of several members of a basin. It was the first described member of the family of chemical compound known as ozonate. Protein has been found to cause Parkinson's disease from both acute and prolonged exposure. Protein is high lipophilic, which penetrates neuron membrane through dopamine transporter. Protein causes aggregates of alpha-synuclein and also inhibition of mitochondrial complex 1 and the impairment of ATP and the release of active oxygen species. Also, protein causes release of cytochrome C, which activates caspase 9 and trigger caspase 3, 6, and 7 and cause cell death. While there is no cure for Parkinson, there are a number of treatments that can ease symptoms. Parkinson medication are the mainstay of treatment. Physical, occupational, and speech therapy can be critical to the treatment plan. Complementary therapies can be used to treat some Parkinson's disease symptoms. Curcumin is a bright yellow chemical produced by Curcuma longa plants. Is the principal curcuminoid of turmeric, another name Curcuma longa, a member of the ginger family Zingibriaceae. It's sold as a herbal supplement, cosmetic ingredient, food flavoring, and food coloring. Curcumin was isolated and synthesized in 1842 from the yellow colored pigment of the turmeric rhizome. Curcuma has been reported to be used in Ayurveda, Siddha, and Unani systems of medicines. It's used as a cooking spice, cosmetic agent, and binding agent. Curcumin has a diverse therapeutic effect in diseases such as liver diseases, lung diseases, neurodegenerative disorder, heart disease, and muscular dysfunction. Curcumin acts on several targets, making this agent able to perform several actions. Niacin. Niacin, also known as nicotinic acid, is an organic compound and a form of vitamin B3. Niacin is essential for humans. It can be manufactured by plants and animals from the amino acid tryptophan. Niacin is obtained in the diet from a variety of whole and processed food. Main functions of niacin: helps convert food into energy, assist in DNA replication and repair. The recommendation: 16 mg for men and 14 mg for women. Good sources: meat, legumes, and cereal. Benefits of niacin: promotes proper function of nerve, improve appetite and digestion, relieve pellagra and skin irritation. Beneficial in treating diabetes, helps to lower blood pressure, aids in conversion of nutrient to energy. Beneficial in curing impotence and erectile dysfunction. Adenosine receptors. Adenosine A2 receptors are located on membranes of several cell types. There are 9 types of adenosine receptor: A1, A2A, A2B, and A3. Each is encoded by different genes. Each type of adenosine receptor has different function. The adenosine A2 receptor is also expressed in the brain, where it has important role in the regulation of glutamate and dopamine release, making it a potential therapeutic target for the treatment of conditions such as pain and Parkinson's disease. Adenosine receptor agonist is used in treatment of Niemann-Pick disease, autism spectrum disorder, and schizophrenia. Adenosine A2 receptor antagonist used in treatment of Parkinson's disease, Alzheimer's disease, Fragile X syndrome, and anxiety. ZM. Example adenosine A2 receptor antagonist. ZM is a high affinity antagonist ligand selected for the adenosine A2 receptor. In animal models, ZM has been shown to protect against beta amyloid neurotoxicity and therefore may be useful as a treatment for Alzheimer's disease. ZM has also been shown to enhance L-DOPA-derived dopamine release and therefore may be useful in treatment of Parkinson's disease. L-DOPA is the most effective standard evidence-based therapy to date. 5 decades after introduction, L-DOPA is still the most effective and widely used drug to alleviate the symptoms of Parkinson's disease. L-DOPA is a precursor to dopamine that crosses the blood-brain barrier and is mainly taken up by the cholinergic neurons that convert L-DOPA to dopamine and increase their dopamine production and storage. L-DOPA is used to increase dopamine concentration in treatment of Parkinson's disease. Side effect of L-DOPA: hypotension, arrhythmia, noisy hair loss, disorientation, and confusion. Aim of the work. The present study. The present study was designed to evaluate the possible therapeutic effect of curcumin and the adenosine receptor antagonist against Parkinson's disease in a mouse model of Rochinon, material and method. Experimental design. Mice were divided into 7 groups. Control group received 20 microliter dimethyl sulfoxide per milli sunflower. Rochinon group received rochinon at dose 1.5 milligram per kilogram body weight subcutaneous every 48 hours for 12 days. Rochinon and the curcumin group received rochinon as in group 2, along with curcumin at dose 80 milligram per kilogram body weight orally. Rochinon and the niacin group received rochinon as in group 2, along with niacin at dose 40 milligram per kilogram body weight orally. Rochinon and ZM group received rochinon as in group 2, along with ZM at dose 3.3 milligram per kilogram body weight intraperitoneal. Rochinon and the curcumin, niacin, and ZM group received rochinon as in group 2, along with curcumin, niacin, and ZM. Rochinon and the L-DOPA group received rochinon along with rochinon as in group 2, along with L-DOPA at dose 10 milligram per kilogram body weight for behavior test is wire hanging test. Wire hanging test is used for daily follow-up the development of Parkinson's disease symptoms as bradykinesia and rigidity. Mice were suspended by its forelimb on a metal rod 40 centimeter length and half centimeter in diameter, located 50 centimeter above the surface. Mice were observed for 60 seconds. The time the mice remained on the rod was recorded. At the end of the experiment, blood was collected and centrifuged to separate serum, and mice were sacrificed by rapid decapitation, and also brain was removed quickly. Parameter in serum, measured in serum, interleukin 6 and angiotensin 2, and C-reactive protein level is used, were measured by ELISA. Parameter measured in brain, adenosine receptor gene, alpha-synuclein level, neurotransmitter levels, noradrenaline, dopamine, serotonin, and acetylcholinesterase levels, oxidative stress marker, glutathione and non-aldehyde levels, and superoxide dismutase activity, lactate dehydrogenase and succinate dehydrogenase activities and ATP level, DNA fragmentation and Cas3 levels, and histopathological examination. Results. Wire hanging test. Rochinon group showed significant decrease compared with control group, while treatment groups with curcumin, niacin, ZM combination, and the L-DOPA showed the amelioration of this reduction. Adenosine receptor gene and alpha-synuclein. And rochinon group showed significant increase compared with control group, while treatment groups showed significant decrease compared with rochinon group. This may be attributed to ZM is selective adenosine receptor antagonist, which produces its neuroprotective through inhibition of alpha-synuclein-induced toxicity. Neurotransmitter levels: dopamine, serotonin, noradrenaline, acetylcholinesterase lipids. This is a significant decrease in dopamine, and serotonin, and noradrenaline levels, and an increase in acetylcholinesterase level in retinol-induced Parkinson group. While in treatment, the groups showed significant amelioration. This may be attributed to curcumin. Curcumin attenuates the sensitivity of T2 receptor to dopaminergic neurons, and also ability to modulate expression of serotonin. Malonaldehyde, superstress marker malonaldehyde, glutathione levels, and superoxide dismutase activity. This is a significant decrease in glutathione level and superoxide dismutase activity, and an increase in malonaldehyde level in retinol-induced Parkinson. While in treatment, the groups showed improvement in glutathione and malonaldehyde levels and superoxide dismutase activity. This may be attributed to niacin decrease in free radical, and also curcumin has a modulatory role on lipid peroxidation and antioxidant result. Lactate dehydrogenase, succinate dehydrogenase activities, and ETB level, and the succinate dehydrogenase and lactate dehydrogenase activities. Endogenous group showed significant decrease compared with control group, while treatment groups showed significant increase compared with endogenous. C-reactive protein, angiotensin 2, interleukin 6 levels. In rotenone group showed significant increase compared with control group, while treatment groups showed significant decrease compared with rotenone group. This may be attributed to anti-inflammatory effect of niacin and curcumin. As B3 and in DNA fragmentation levels, in rotenone group showed significant increase compared with control group, while treatment groups showed significant decrease compared with rotenone group. This may be attributed to curcumin reverse inhibition of DNA repairing enzyme. Pathological studies. Photomicrograph hematoxylin and leucine of the hippocampus. In control group showed the normal histological feature, well-defined molecular and granular and normal neuron cell. And rotenone group showed the abnormal histological feature represented by vacuolization and edema of neuron cell. In treatment groups showed the almost normal histological feature, well-defined molecular and granular and normal neuron cell with visible axon, and some neurodegenerative change as vacuolization were also seen. Thus,
previous hypothesis. Yeah, previous.
Photomicrograph hematoxylin and leucine of the substantia nigra. The effect of substantia nigra is the same shown in the hippocampus. Conclusion and recommendation. Curcumin and niacin may be considered as promising food supplements for the management of Parkinson's disease. Treatment with L-DOPA and the adenosine receptor antagonist ZM had comparable ameliorating effect. The combination of ZM with niacin and curcumin produced the most potent improvement effect. More studies on ZM, curcumin, and niacin are needed to further assess their efficacy as a promising agents in the management of Parkinson's disease. Thank you.
صباح الخير. Studying immune reprogramming associated with changes in carbohydrate metabolism upon influenza infection using an in vitro cell cultured immortalized macrophage system. دراسة التغيرات المصاحبة لإعادة البرمجة المناعية في أيض الكربوهيدرات الناجمة عن عدوى الإنفلونزا باستخدام نظام معملي مبني على مزارع الخلايا المناعية الأكلية. صباح الخير. The presented work was submitted for the partial fulfillment of the degree of PhD in pharmaceutical sciences in biochemistry by Karim Awad under the supervision of Professor Dr. Tariq Kamal Mutawa, Professor of Biochemistry, Faculty of Pharmacy, Cairo University, Professor Dr. Mahmoud Bahgat, Professor of Immunology and Infectious Diseases, Division of Pharmaceutical Industry, National Research Center, and Professor Dr. Amani Said Mawrabi, Professor of Immunology, Division of Pharmaceutical Industry, National Research Center, Associate Professor Dr. Nancy Nabil Shaheen, Associate Professor of Biochemistry, Faculty of Pharmacy, Cairo University, and as internal monitor, Associate Professor Dr. Tina. The presented work also has been published in EUPMB Life under the title Glycolytic and Immunological Alterations in Human Immunoglobulin 3/7 monocytes in response to H1N1 infection, and in viral immunology under the title H1N1 infection reduces glucose level in human immunoglobulin 3/7 monocytes culture. This talk will be presented in this order. First, an introduction on H1N1 virus, immunology, and the accompanying metabolic changes. Then the aim of the work, the design of the experimental work, results, conclusion, and finally, key messages and recommendation. H1N1 influenza virus. Influenza is an acute respiratory disease that can cause local annual epidemics and worldwide pandemics of different morbidity and mortality. When infections with influenza A virus were reported in Mexico and the United States in 2009, the virus rapidly spread worldwide, where eventually more than 214 countries and overseas territories reported laboratory confirmed cases that created the 2009 WHO reported pandemic, including more than 18,000 deaths. Since then, H1N1 virus became a major public health problem around the world. This map shows the number of confirmed cases around the world during the peak of 2009 pandemic. Egypt was among the first category with more than 50,000 confirmed cases. The influenza virion or infectious particle is a spherical shaped enveloped virus with single-stranded RNA and an outer layer membrane derived from the host cell in which the virus multiplies. The three proteins form spikes on the surface of the virus, hemagglutinin, neuroamidase, and the M matrix proteins. Those are responsible for determining the type and subtype of the influenza virus. Hemagglutinin and neuroamidase are responsible for infection. Hemagglutinin is responsible for attachment of the virus to the host cell, and neuroamidase is responsible for eating of sialic acid from glycan of the infected cells. Viruses can attack both somatic cells and immune cells. When they attack somatic cells, virus products or type 1 interferons are released, which act on both adaptive and innate immune cells. They act on the adaptive immune cells to release specific antibodies, and on the innate immune cells to introduce these infected cells to the innate immune cells to engulf them or destroy them. When they attack immune cells, specific inflammatory mediators are released, which act in different manners. For example, TGF-beta may act on the blood vessel wall to produce plaque formation and blood clotting. Also, TNF-alpha may produce filial hyperplasia. Toll-like receptors. pathogens are identified through recognition of certain molecular structures or surface receptors that are absent in uninfected cells, termed pathogen-associated molecular pathogens. Two types of pattern recognition receptors, which are toll-like receptors and retinoic acid-inducible gene 1 like receptors, which signal viral infection and then activate immune cells to produce specific markers. Each pattern recognition receptors depends on the cell type and the nature of the pathogen. In regard to their localization, toll-like receptor 3 and toll-like receptor 7 are located in cell compartments and are released upon viruses or RNA viruses infection. So manipulating toll-like receptors can be a promising approach, which could lead to localization of the virus or the virus products at the site of infection. Metabolism in immune cells during infection. Metabolic pathways in immune cells are modulated either through interactions with metabolic signals from the affected organs, such as diverse conditions of nutrients and oxygen availability, or co-effectors in their macro and microenvironment, like infection products, or chemicals. The need for energy for such cellular functions is essential, with glucose metabolism being the major carbon fuel source through two distinct pathways: glycolysis at the first pass producing pyruvate and ATP, and the tricarboxylic acid cycle as an immediate following pass. Glycolysis is regulated mainly by the three enzymes: hexokinase, phosphoruptokinases 1, and pyruvate kinase. Phosphoruptokinases 1 is the rate-limiting enzymes in glycolysis. Among the metabolic enzymes that also regulate glycolysis, phosphoruptokinases 2, a family of bifunctional enzymes that catalyze the formation of fructose 2,6-biphosphate. So phosphoruptokinase 2 allosterically modulates phosphoruptokinase 1. So a correlation may exist between the concentration of phosphoruptokinase 1, phosphoruptokinase 2, and the rate of infection. The concept of trained immunity or innate immune memory or immune reprogramming. The immune system has evolved to protect against pathogenic microbes while avoiding harming self-tissues. The fact that the adult immune system could be tolerated to non-immunogenic forms of antigens was a key discovery that was to shape our thinking on how to reprogram the immune system to establish tolerance therapeutically. Long-term changes in innate immune cells can be achieved by manipulation on the epigenetic level instead of producing permanent genetic changes. This long-term change leads to an increased responsiveness upon second stimulation by microbial infection. So the cells involved in the trained immunity is innate immune cells like macrophages, and the mechanisms include cell physiology and the role of transcriptional factors. But of course, had a therapeutic values when considering the production of non-specific vaccines or the well-adaptive innate vaccine products, and the maladaptive states could be a shifting metabolism towards a high glycolysis or high metabolic state of the cell. U937 cell lines as a model system for this study. U937 is a human hematopoietic cell line with exceptional characteristics derived from the patient with generalized histiocytic lymphoma. U937 being a pro-monocytic cell line can be differentiated in vitro into either various types of macrophages or dendritic cells in response to different stimuli. These cells have become a diverse continental model in studying different parameters, including glucose metabolism. In the word, the present study aimed at studying the carbohydrate metabolic changes occurring in cultured immune cells, U937 monocytes, upon infection with influenza virus, and the subsequent immunological responses. Addressing the correlation between sugar kinases, phosphoruptokinase 1 and phosphoruptokinase 2, and cytokines, as well as toll-like receptors regulated upon influenza infection to enable better understanding of factors involved in human host susceptibility or resistance to infection. source of U937 cells and H1N1 virus used in this work. The Puerto Rican H1N1 virus was prepared by Plasmid Rescue System and provided by Professor Rigert Whippy from St. Jude Children's Research Hospital in USA through a signed material transfer agreement with the National Research Center. The U937 cell line was provided by Professor Dr. Michael Kirschfink from the Institute of Immunology of the Medical Faculty in Heidelberg. In this work, for each 10 to the power 6 cells, 10 to the power 5 virus particles were used for infection, and this provides a multiplicity of infection of 0.1. We adopted in this study 3 types of U937 cells. The pathogen-stimulated ones, either by live or killed H1N1 or staph virus bacteria. The non-pathogen stimulated one by the polymer state acetate or human serum, and the unstimulated cells. We subjected all to infection with H1N1 at a multiplicity of infection of 4.1. And we gathered the supernatant and cell lysates at different time points, 12, 24, and 36 hours. We measured the parameters phosphoractokinase 1, phosphoractokinase 2, TNF-alpha, toll-like receptor 3, and toll-like receptor 7 on the genetic level by the qRT-PCR and on the protein level by ELISA. We also measured phosphoractokinase 1 activity and the glucose in the culture media and the pyruvate concentration, all calorimetric. Phosphoractokinase 1 enzymatic activity in the lysates or membrane unbound of an uninfected or H1 infected U937 cells. This figure shows phosphoractokinase 1 activity in infected H1N1 or BMA stimulated cells or infected unstimulated cells compared to the uninfected cells. Significant increase was observed in infected H1N1 or BMA stimulated cells or infected unstimulated cells compared to the uninfected unstimulated cells and compared to the corresponding uninfected cells at the 3 time points. Similar results were observed for phosphoractokinase 1 enzymatic activity in the pellets or membrane-bound phosphoractokinase in uninfected or H1 infected U937 cells. Significant increase was observed in phosphoractokinase 1 in the extracted pellet. And H1N1, the infected H1N1 stimulated or BMA stimulated or unstimulated infected cells compared to the uninfected unstimulated cells and compared to the corresponding uninfected cells. Pyruvate production lysates after 12, 24, and 36 hours. This figure shows pyruvate concentration in the infected H1N1 stimulated or BMA stimulated or infected unstimulated cells compared to the uninfected unstimulated status and the corresponding uninfected cells. We observe fluctuation in the pyruvate concentration. Either significant or non-significant increase was observed for BMA stimulated infected cells and infected unstimulated cells compared to the uninfected cells and the corresponding uninfected stimulated cells at the 3 time points. Also significant or non-significant increase was observed in infected H1N1 stimulated cells compared to the corresponding uninfected cells after 24 and 36 hours. All suggest high energetic cells in the context of glycolysis. TNF-alpha detection in the lysates of uninfected or infected H1N1 U937 cells by ELISA. This figure shows TNF-alpha level in the lysates of infected H1N1 stimulated or BMA stimulated or unstimulated infected cells compared to the uninfected cells. Significant increase was observed in TNF alpha concentration in H1N1 in infected H1N1 stimulated cells or BMA stimulated cells or unstimulated infected cells compared to the uninfected unstimulated cells, also compared to the corresponding uninfected stimulated status at the 3 time points, which suggests enhanced immune responses due to infection. Toll-like receptor 3 detection in the lysates of infected or uninfected H1N1 U937 cells by ELISA. This figure shows toll-like receptor 3 level in in infected H1N1 or BMA stimulated cells or unstimulated infected cells compared to the uninfected unstimulated cells or the corresponding uninfected cells. Significant increase was observed in H1N1, infected H1N1 or BMA stimulated cells or unstimulated infected cells compared to the uninfected unstimulated cells at the 3 time points. However, significant decrease was, or non-significant decrease was observed compared to the corresponding uninfected stimulated status. Toll-like receptor 7 detection in the H1N1 infected or uninfected U937 cells by ELISA. Like TNF alpha, toll-like receptor 7 shows significant increase in H1N1, in infected H1N1 or BMA stimulated cells or unstimulated infected cells compared to the uninfected unstimulated status at the 3 time points. Also, significantly increased compared to the corresponding uninfected stimulated status. All suggests enhanced immune responses during infection. Gene expression of inflammatory cytokines in the lysates of infected or uninfected U937 cells by qRT-PCR. This figure shows the inflammatory cytokines TNF alpha, interferon beta, and interferon alpha relative mRNA expression in the infected, stimulated or unstimulated cells compared to the uninfected, unstimulated cells. A generalized reduction was observed in all infected or stimulated cells compared to the unstimulated, uninfected cells. That was on contradiction to the protein concentration, which may suggest the contribution of epigenetic modulators or factors. Toll-like receptor 3, toll-like receptors 3 and 7 mRNA relative expression in uninfected or infected U937 cells by qRT-PCR. Also, a generalized reduction in all stimulated or infected cells where it was observed compared to the uninfected, unstimulated cells. Similar observation was observed for phosphoruptokinase 1 and phosphoruptokinase 2 concerning the generalized reduction of all stimulated or infected cells compared to the uninfected, unstimulated status, with only one exception here. 3 isoforms were identified for phosphoruptokinase 1, phosphoruptokinase 1 muscle, platelet, and liver. We have been observed that phosphoruptokinase 1 mRNA expressed in monocytes was in profile similar to phosphoruptokinase 1 in liver, even if less in intensity. This figure shows the correlation between the studied inflammatory cytokines and phosphoruptokinase 1 concentration. The heat map gives an evidence of trust in their correlation. It summarizes the correlation between all studied molecules at both the transcriptional and the translational levels at various stimulation and infection condition. This figure shows that phosphoruptokinase 1 protein concentration positively correlated with protein with TNF alpha and toll-like receptor 7 protein concentration post-infection, negatively correlated with toll-like receptor protein concentration, also negatively correlated with TNF alpha and toll-like receptor 3 and toll-like receptor 7 mRNA relative expression. Glucose profile in the infected and uninfected cells. We have observed the reduction in the glucose concentration in the culture media of infected cells compared to the uninfected cells, or in unstimulated cells, also in stimulated cells either with live or killed virus or live or killed bacteria. The same reduction was observed when cells were stimulated with either BMA or human serum. All evidences the utilization of glucose by the virus. Conclusion: H1N1 infection modulates glycolysis in human U937 cells. We have observed a significant increase in both soluble and membrane-bound phosphoruptokinase 1 activity in infected cells compared to uninfected cells, regardless of the previous stimuli. In spite of decreased or increased pyruvate profile in infected cells compared to uninfected cells in regard to the study time points of initial cell stimuli. H1N1 infection significantly reduces glucose level in U937 culture medium. H1N1 infection stimulates immune responses in human U937 cells. We observed increased TNF alpha and toll-like receptor 7 proteins in infected cells compared to uninfected cells, regardless of the previous stimuli. Toll-like receptor 3 protein increased in infected cells compared to the uninfected unstimulated cells. However, decreased in H1N1 or BMA cells compared to their uninfected stimulated status. This study reports discrepancy between the protein markers and their genetic expressions upon H1N1 infection. So we observed increased concentration of TNF alpha, toll-like receptor 3, toll-like receptor 7, and phosphoruptokinase 1 proteins in infected cells compared to uninfected cells, unlike decreased expression of these markers in mRNA infected cells compared to uninfected cells. This study evidences statistical correlation between the studied immunological and metabolic parameters. Key messages: as non-living antigens, viruses don't inherently have their own metabolism. Rather, upon infection, they dramatically alter the metabolism of the host cell. These alterations of the host cell metabolism can provide the substrates necessary for viral replication. Viruses depend on the host metabolism in order to perform the necessary tasks leading to the replication process, which requires a high amount of energy in a short time. The experiments have demonstrated type and time-related metabolic patterns in infected cells that might be related to different responses when it comes to virus type specificity or the infection dynamics. And recommendations. This study recommends U937 cell line as a serial model to screen immunomodulatory and biochemical agents regulating influenza virus infection. Further studies on association between metabolic kinases and immune cytokines are required in other immune cell lines and primary human immune cells. Further studies are also required to investigate acting epigenetic factors in this crosstalk between metabolic kinases, inflammatory cytokines, and toll-like receptors. Finally, since glucose concentrations in U937 cell cultures are not constant following H1N1 infection, time course monitoring of glucose concentration in experiments done in vitro cell culture model infection is recommended.
بسم الله الرحمن الرحيم. السلام عليكم مرة ثانية. أنا في نهاية ورشة العمل النهارده أو الورك شوب لا يسعني غير إن أنا أشكر السيدة الأستاذة الدكتورة الفاضلة منال عبد العزيز حامد رئيس شعبة الصناعات الصيدلية والدوائية على دعمها والحقيقة وتشجيعها على إن إحنا نعمل ورشة العمل دي برغم ضيق الوقت وبرغم إن الفكرة اتولدت بسرعة. وطبعا انا عايز اخص بالشكر السيده الاستاذه الدكتوره الفاضله والزميله العزيزه الدكتوره هبه الرفاعي وكيل الشعبه وبصفتها مشرف على وحده المؤتمرات، الحقيقه لانها ذللت كل العقبات وحطت معانا الاستاف بتاعها كله عشان تدعم ان ورشه العمل دي تحصل، برغم حضراتكم عارفين يعني ان الوقت ضيق قبل اجازات العيد. أنا عايز أقول حاجة مهمة جدا. الهدف من ورش العمل إن إحنا كنا ندي فليفر، نوع من أنواع التيستنج، ندي فرصة للناس تتيست الفليفر بتاع الريسيرش الأونلاين ريسيرش أكتيفيتيز اللي موجودة في قسم الكيمياء العلاجية في شعبة بحوث الصناعات الصيدلية والدوائية. كان في 2 ديزيز مودلز النهارده اتكلمنا عن المانجمنت بتاعهم او الترايلز بتاعتنا تكونتريبيوت تو ذا مانجمنت، مش هنعرف نعمل كل حاجه لوحدنا. واحد منهم كرونيك نيورو ديجنريتيف ديس اوردر باركنسون، واحد منهم انفلونزا انفكشن ريسبيراتوري انفكشن، وخدنا معاه كمان اون توب سكندري بكتيريال انفكشن ستاف اوريوس. واتكلمنا على ان احنا نعمل برايمري اميون سيل كلتشر موديل عشان نريبورت على بروتين اكسبريشن بروفايل وجين اكسبريشن بروفايل. ده كله حقيقه could be happened على ارض الواقع في المعمل عندنا وفي القسم عندنا او في معامل القسم. الحقيقه ما اقدرش اقول معمل واحد لان هم 8 معامل الحقيقه بلس معامل ممثله في مراكز تميز. ارجو ان ده يبقى تريجر كويس ان احنا نبتدي نتعاون مع مجموعات واقسام اخرى في المركز القومي للبحوث. ده دايما في انكارجمنت من رئاسه الشعبه ان احنا نعمل حاجه زي كده. كل سنه وحضراتكم طيبين واتمنى لكم اجازه عيد سعيده ان شاء الله. شكرا جزيلا.